Okay, hi everyone. I'm Rebecca. I'm a first year CBM student, and I bravely did a long read sequencing project, a spatial project. Okay. So I think we're all familiar at this point with the many modifications that are added to RNAs, including methylation, pseudouridine modifications, and so on. And these modifications are crucial for regulating various processes in the cell that involve RNAs. And they're present not only on mRNAs, but also on tRNAs and ribosomal RNAs and other types of RNAs. And something actually that I learned while I was preparing for this presentation is that it's kind of interesting. RNA modifications can be one of the cell's first lines of defense in responding quickly to an external stimulus, because the cell can upregulate enzymes that deposit or erase these modifications, and the cell can respond more quickly in that way than changing their transcriptome or their proteome. So yeah, a really crucial aspect of cellular regulation. We're also by now very familiar with the tech being developed by Oxford Nanopore Technologies, but just as a refresher, a huge advantage of the pore-based sequencing is that as the DNA or the RNA molecule moves through the pore, you can detect not only the base, but if there are any modifications as well, because it alters the electrical signal. And one setting where it's really interesting to study these modifications is in the context of viruses, where it's well known that RNA modifications are crucial for the viral life cycle of RNA viruses. But even less is known about the types of modifications that are present or what they do. So, and then of course, there's a therapeutic aspect to this because the more we understand how these viruses work, the more opportunities that opens up for potential intervention. So I analyzed some data from long read sequencing of the hepatitis C virus to see what RNA modifications I could detect. And I had two data sets, one where the RNA from the virus was generated via in vitro transcription. So that's our negative control. It should have no modifications. And then I also had data from a total RNA sequencing experiment from cells that were infected with hepatitis C. So that's where we're hoping to see some actual modifications. And if the data looks familiar, that's because it was the same data set that we worked with for homework number 2. So a new version of the Dorado base caller was just released that has some new base calling models. And it allows for detection of a new RNA modification called 2' O methylation. So this can occur on any base, and it's a really abundant and also highly conserved modification, especially on ribosomal RNA. So I was excited to use the latest version of the Dorado base caller to also include these modifications in my analysis. Yeah, highly abundant on ribosomal RNAs. So I ran the latest version of Dorado on the hepatitis C virus data with all the available base calling models, and this is what I was able to generate. So the x-axis here are all of the modifications that I performed base calling for, and the y-axis are the total number of modifications that I detected for each sample. And I'm only considering the mapped reads for this plot. And you also have to remember that the total RNA sample, if you'll remember from our homework, it had more mapped reads than the negative control, the in vitro transcribed sample. So that's the reason why there are so many more modifications detected in the total RNA, on top of the fact that that's the sample where we expect to actually detect, real modifications that are not artifacts. I also plotted the pass-frac column, which is the fraction of modifications that passed the threshold I was using, which was 0.95 for all of the modifications. And you can see from the y-axis there that only a small fraction of the modification calls were passing my filter, but I guess I was using a pretty stringent threshold. I think for the purposes of this analysis, I was just trying to see if I could detect any sites that we could say with high confidence were probably modified. So then I tried to look for sites that were modified in the total RNA sample, but not in our negative control, since these are the sites that are most likely to represent true modifications as opposed to artifacts. So I took all the modified sites and I applied the filtering requirements on the left. So I required coverage of at least 2 molecules for each site in both of the samples, and then at least 2 of the molecules had to be modified in the total RNA sample. So basically, I didn't want to rely on a single molecule to support the site in either sample. And then I also lastly required that none of the reads had a modification in the IVT sample or negative control, which sounds a little stringent, but the coverage was pretty low in the sample. So it usually only had 1 or 2 molecules covering a site to begin with. And then on the right, you can see the number of sites for each modification that passed these thresholds. So there were a handful of M6A sites in blue on the left that passed these criteria. There were also a few 2'O-methylguanosine sites, which is one of those new modifications that we can only see with the new base color, and that's in purple. I also saw some variation within the same modification depending on the model that I used. So for example, if you look at those 2 blue bars, I saw 5 sites meeting this criteria with the original inosine M6A model, but then 12 sites that passed if I use the newer model that can also call 2'O-methyladenosine as well. So it's been mentioned before in the class, but just something to be aware of if you're working with the Dorado base colors, potential variability between the different models when they release updates. And then lastly, I just highlighted a couple of example sites. So this first site that passed my filtering on the top, it passes differentially modified site for M6A. And the total RNA sample had a coverage of 161 molecules at this site, and 7 of them were modified. Meanwhile, in the in vitro transcribed sample, we had a coverage of 2, and neither of the molecules were modified. So this is kind of the most convincing evidence that I could get of a modification. And you can see similar evidence of an M5C modification site, as well as a 2'O-methylguanosine modification. So it does seem like we have some evidence that we can detect these modifications in the total RNA sample. So in conclusion, we were able to detect evidence of at least a couple of modifications in the total RNA sample for the hepatitis C virus, using the latest version of the Dorado base color allowed for detection of these 2'O-methylguanosine modifications. But sequencing depth of both of those samples limited the detection capability. And if I had more time, I would probably start looking at where these differentially modified sites were and trying to understand if there are any biological interpretations you can make of them. But yeah, any questions?